Hello everyone, I am Professor Ronika and today I will be presenting on a fundamental piece of laboratory equipment, the pH meter. We will cover the core concepts, the detailed working principle, its essential components, the steps for accurate measurement, different types and its wide range application across various industries. First, what exactly is pH? pH is the measure of the acidity or basicity of an aqueous solution. Mathematically, it's defined as the negative logarithm of the hydrogen ion concentration. This logarithmic scale compresses a huge range of hydrogen ion concentrations into a simple, manageable scale from 0 to 14. 0 is highly acidic, 14 is highly basic or alkaline, and 7 is perfectly neutral like pure water. Now to measure this value accurately, we use an electronic instrument called the pH meter. As you can see in the image, it's an electronic instrument that consists of a probe, usually a combination electrodes, connected to a meter. It does not use chemical indicators like litmus paper. Instead, it measures the electrical potential difference between two electrodes submerged in the solution, which brings us to its operating principle. The pH meter operates on the principle of potentiometric measurement. Essentially, it functions as a tiny electrochemical cell. The key component here is a specialized thin glass membrane at the tip of the electrode. This membrane is highly sensitive to the concentration of hydrogen ion both inside and outside the electrode. When the electrode is placed in a solution, the difference in hydrogen ion concentration on either side creates a tiny voltage or electrical potential across the glass membrane. The meter measures this voltage difference, and that potential is mathematically linked to the pH by the nearest equation. This equation, which you see here, shows that the measured potential E is directly dependent on temperature and the activity of the ion being measured. The pH meter unit uses this relationship to convert the measured voltage directly into a readable pH value. This explains why temperature is so critical. The measurement begins at the electrode. Modern pH meter almost always use a combination electrode. As the name suggests, it houses two electrodes in a single compact body. It contains the glass electrode or the indicator electrode. This is the hydrogen sensitive component containing a silver chloride wire and an inner buffer solution, typically pH 7. The voltage develops across its glass tip. The second one is the reference electrode. This provides a stable, known and constant potential that acts as the baseline for the measurement. It uses a small junction to connect with the sample solution. The meter measures the voltage difference between these two internal systems. The whole unit is designed to be highly sensitive and highly resistant, which leads us to the meter unit itself. The electronic meter unit is the brain of the operation. It has four critical functions. First, the high input impedance amplifier. The voltage generated by the electrode is extremely small and the glass membrane has very high resistance. This amplifier measures the tiny voltage without drawing any significant current, which is absolutely crucial for an accurate reading. Second, the digital display, which simply shows the resulting pH value and often the temperature. Third and very important is automatic temperature compensation or ATC. Since the Nernst equation shows pH is temperature dependent, the ATC probe measures the sample temperature and automatically adjusts the meter's calculation. This ensures the pH reading is accurate regardless of the sample temperature. And finally, the calibration control used to adjust the meter using a known buffer solution to ensure accuracy. Calibration is the most crucial step for accurate pH measurement because the electrode's potential naturally drift over time. We perform a 2 or 3 point calibration using certified pH buffer solutions. Step 1 is rinse. We always start by thoroughly rinsing the electrode and the ATC probe with deionized water to prevent cross-contamination. Step 2 is natural buffer. We immerse the probe in the pH 7 buffer. The meter measures the voltage at this 0 point and adjusts the reading to exactly 7. This is the offset adjustment. Step 3 is second buffer. We rinse again and then immerse the probes in a second buffer, typically pH 4.01 for acidic samples or pH 10 for basic samples. This step measures the slopes of the electrodes, how much the voltage changes per pH unit. This ensures the meter responds correctly across the pH range. The third buffer is often used for maximum accuracy across the full acidic and basic range. Now the procedure for sample measurement. Once the instrument is calibrated and the electrode slope is verified, we are ready to measure the sample. The step first is rinse and condition. We rinse the electrode again. It's also good practice to give it a quick rinse with a small portion of the sample itself to condition the glass membrane and prevent dilution. Second step is immerse and stir. Place the electrode and ATC probe deep enough into the sample. Gently stir the solution to ensure homogeneity and proper sample flow near the junction. Do not stir vigorously. Then the third step is stabilization. Wait patiently. You must allow time for the electrical potential to stabilize. Look for the ready indicator on the meter or wait until the number stops drifting and then record the stable pH value and temperature. Finally, and critically important for electrode lifespan, the storage. The electrode should never be stored in pure deionized water, as this damages the membrane. It must be stored in a specialized storage solution, typically a potassium chloride solution. Now the types of pH meters. pH meters come in various designs to suit different application. The first one is benchtop meters. These are the standard laboratory grade instrument we have discussed, offering the highest accuracy and advanced features like data logging and multiple collaboration points. The second type is portable or handheld meters, or battery operated, designed for field use such as environmental testing, often built to be durable and waterproof. Pocket testers or pH pens are the simplest pen-sized versions used for quick generally measurements where high precision is not required. The choice depends entirely on the required accuracy, portability, and the environment of use. The pH meter is essential in almost every scientific and industrial fields. In chemistry and biology, it's used to prepare stable buffers, monitoring the progress of chemical reactions and maintain the correct conditions for cell culture media and protein stability studies. In environmental monitoring, we use it to test for acid rain and monitor ocean acidification, which is critical for marine ecosystem. In water treatment, it ensures our drinking water and wastewater effluents meet strict pH regulatory standards. The food and beverage industry relies on it heavily, controlling fermentation for beer and wine, monitoring milk quality, and ensures the correct acidity for food preservation and canning. Even in agricultural and soil science, pH meters are used to test soil, determining the optimum fertilizer and crop for a given area. To give you a better sense of scale, here are some practical examples. A highly acidic substance like battery acid has a pH near 0. Something mildly acidic like coffee is around pH 5. Pure water is neutral at pH 7. Baking soda solution is mildly basic around pH 8.5. And highly basic substance like household bleach can reach pH 13. These examples underscore why accurate real-time measurement is necessary for quality control and scientific inquiry. To conclude, the pH meter is an indispensable tool. It works by measuring the electrical potential differences between the glass and reference electrodes, a relationship governed by the Nernst equation. Remember that calibration with standard buffers is mandatory for accurate results, and automatic temperature compensation is crucial due to temperature sensitivity. Whether in a research lab or water treatment plant, pH meter plays a vital role in ensuring quality, safety, and scientific precisions. Thank you very much for watching.